Hi, this is Dr. Ben Morrill. Welcome to episode 41 of Reptile Genetics Weekly. Got some cool stuff to talk to you about, some great successes we've been having these last few weeks. It's been really nice to see things coming together, getting faster and more tests both at the same time. So as far as updates for shed testing, we have right now the panels and the fast testing I'll get to in a second, but the panels and everything else that's not on fast testing yet, those we'll do that big run the first week of February. So I guess it's only like a week away. We'll start that run. So it'll probably be the second week in February that we'll have results. But as far as the fast testing, we have another big batch of results going out today. Thanks to all of you that are sending samples in. We got quite a few just when we're just doing them like every other day. We're still getting quite a few sheds coming in, so that's awesome. And shout out to Showcase Serpents. They sent quite a few and got some cool results for you. But yeah, those will probably go out either this afternoon or this evening. So if you send in for Clown, DGC, or VPI, those are going out today. And then we'll have more go out in a couple days. So we'll have them going out pretty much 2 or 3 times a week. So they're coming, like we said, 2 business days. And the other cool thing, well, I guess we'll go ahead and bring Kayla on before we talk about the rest. Kayla, how are you doing?
Hey, I'm doing good. I just can't get over how awesome it is that you guys can send out results that fast.
Yes. It feels so much better. I always felt so bad when results took a long time. Every single one I felt so bad. This feels so much better. Like get results, do the run, have results, get them sent out all the same day, just like
that. Yeah, no, it's
do a run every other day or whatever. It's really nice.
Yeah, that's wild. Just to hear, okay, they'll be out in like 2 or 3 days. Like, whoa. Well, actually, no, less than that. 1 or 2 days.
Most of the time, it should be 1 to 2. Yep, 2 days or less.
Definitely. So we started this on the 15th, and here we are, I guess, a week later now. And yeah, so the first ones that we rolled out were Clown, DGC, and VPI Xanthic. But apparently, Ben, you've been working on something else, right?
Yeah, we've got 3 more working. So we've got Pied and Puzzle and Sunset. All 3 of those are working. So I'll have to scale those up. So each time I get one working, I have to scale it up so that we can, have it available and ready for people, for customers. So it'll be about 2 weeks until we'll have these 3 where we're able to do them every other day, like we're doing with Clown, DG, and VPI. But yeah, these 3 are working. They're beautiful.
Love to hear that.
Yes.
Also, honestly, it surprised you and me. Like, and I knew you were working toward more, but it surprised you and me that you had these 3 out already. Yeah. So or on the way to being out. So that's cool.
Yep. And I expect in the month of February, we'll have a dozen more that will be working.
Oh, geez.
Real quick.
That'll be great.
It's been burning pretty long hours, but it's been paying off. It makes the long days and nights a lot of fun when stuff keeps working. So
yeah, definitely
don't mind at all when it keeps working. So yeah, we'll have a whole bunch more. We'll have those 3 come online in 2 weeks, and then another 2 weeks after that. Hopefully we'll have another 6 to 10, and by the end of February we should have close to a couple dozen that are all switched over to the fast testing.
All right, well, that'll be fun. So for those of you who don't know, we do have a sale going on for our Blue-Eyed Lucy complex. You might have seen it on our social media. If you got a Blue-Eyed Lucy, you don't know what flavor it is, this is how you find out. And for way cheaper than usual.
Yes.
And likewise for the YB complex, we are running those. And apparently, Ben, you're working on getting those into a fast test as well, right?
Yeah. We should be able to have Blue-Eyed Lucy and Yellow Belly complexes as part of our fast testing over the next. I don't know if we'll get all, because that's 12 different tests. I know most people know Yellow Belly or the Yellow Belly complexes. They think of that as being 5, but for Yellow Belly, we have to run 2 tests. So that's 12 different tests I have to get switched over. But give me a month or 2. We'll probably have both of those going as fast tests as well.
All right, that'll be good. Well, I flipped the screen a little early there, but so I kind of gave it away. But why don't you tell us about Late to the Mic?
Yeah, I just wanted to mention. So this is Patrick and Troy and Chase. So Patrick from Chimera, Troy from Best Dressed Balls, and Chase from Canova. They have a cool show that they're doing each night. It's pretty late on Thursday nights. I want to say it's 10 p.m. Eastern, but I'm not totally sure.
Yeah.
But anyway, they've had some really cool guests come on lately. They had Justin Kabelka last week, and they've got Emily and Ed from Snake Discovery that are coming on this next, in a couple of days.
Oh, that'll be cool.
It's pretty cool. It's a nice late night light. It is adult language, so it's a little different from our show. So you will hear some adult language. But I really like the format, what they're doing, and the people they're bringing on, and so it's cool. I just wanted to give a shout out and give a heads up if you're okay with adult language, don't have kids around, small kids at least.
Yeah.
It's a fun show to listen to.
Well, sounds good. I know I'll be giving it a listen. It looks like a good crew. And also I recognize all of those names in that little, like, those are their last 4 videos I got a screenshot of, and it looks like fun.
Yes.
Yeah. So this is one of our Got Shorty Sheds, or Got Sheds as I call them, social media posts that you'll see occasionally on our Facebook and Instagram. And basically it's just a call for different parts of the community to send in sheds from different species. So what we're going to do now is basically we're just going to ask Ben where we're at with getting sheds for different species.
Yeah, so we've had quite a few people send in sheds for short tails and bloods. So thank you. We can definitely still use more. And people ask, what morphs do you still need? All of them.
All of them.
I have a decent, I probably have the most T negative, but I still could use more, especially if they're homozygous, if they're visual T negatives. I can still use more. So any of them, send them all in. Those will be helpful. One of the biggest reasons, and we mentioned this in the live, that we're bringing this up right now is because when I went and gave a talk at the genomics conference last week, I guess it was, made some more good connections and had some good conversation. And so this is looking like we will be doing this soon. So we'll be able to spend the money and get some deeper whole genome stuff done so we can do more mapping. So yeah, that's another reason to send sheds in. And also while we're going through these, for any of these species that we don't have a good whole genome for, we will need to collect blood samples just from like 1 or 2 individuals. So if any of you that are watching this are somewhere, like I know we have a couple people that are at the University of Florida that have the access to be able to get blood draws from snakes, which is not easy to do. I've done it a few times when I was a grad student. It's not simple. Someone needs to have experience. If you call your local vet and they say, "Oh yeah, we can do that," say, "How many times have you done it?" Because if they've never done it, they're not going to be able to get it, most likely.
Wow.
It's not easy. Really? So yeah, if any of you watching happen to be like, at a vet university, or have some kind of access, or are a veterinarian or work for a veterinarian that has reptile experience, that's one thing we'll need to get the real high quality reference genomes to be able to map to. We need blood samples. So just shoot us a message either through Facebook, Instagram, YouTube, any of those. Just let us know, and the more options we have, the faster we can get the samples we need to make this happen.
Yeah. And just out of curiosity, do the blood samples need to be from normal animals, like wild type, or can it be from a morph? Does it?
Ideal would be a wild caught animal with, complete normal genetics. So when we did the only one we've paid for and done ourselves is the ball python genome that we use to do all of our mapping. And we actually got a male and a female. And I was just sharing a cool story with Kayla that we'll show here in a few episodes, I hope.
Oh, I'm so excited for that. It's cool.
Yeah. So we have good pictures and story where these wild caught ball pythons, pictures of them, where they were caught, and cool story with them. So Steve Hillis is one of the ones that's at University of Florida. he's the one that collected them and got the blood samples to us. So
Very
that's
nice.
cool when we can show that. But yeah, the ideal would be wild caught with location information, like GPS coordinates. That would be awesome. But if the best we can do is something that does have a morph or two, then we can make it work. But the more wild type, no captive breeding at all, the better.
Yeah, makes sense. All right. So where are we for Hognoses?
So Hognoses, we've had a lot of really good samples sent, like good numbers of samples sent in. I would still say with any of them we're going to talk about, I would say there's not any that I have too many of yet. So if you have hognose morphs, definitely we can still use all of them. But hognose keepers, you've done a good job of getting stuff sent in. We appreciate it. And that's moved hognoses, probably hognoses and boas are at the very top of our list as far as a brand new genome. Blood pythons and retics and Burmese, I'm hopeful we can use our ball python genome to do mapping for them. But as far as like brand new genomes that at least I haven't worked with at all yet, hognose and boas are probably at the top of our list.
Very nice. Good job. I actually didn't realize that the boa community had stepped up like that. I think they're okay. They're not the next slide, but retics. Yes. I know that would be really nice if you're able to save yourself some of the work by using the ball python genome to map out retics.
And we'll just have to see when we try. It might work okay or it might not. So
yeah, fair enough. Oh, I hear your phone. I don't have a berm slide, but I'll ask for them. How are they looking?
Very few, but I do have a few.
Okay.
Definitely could use lots more. Same with retics. Well, there's more retic samples that I've gotten than Burmese, but yeah, definitely still send them in.
All right. Let's see. Boas, you mentioned that you have a decent amount of them. Any particular morphs are there any morphs that you're satisfied with, or do you still want more of everything?
No, we definitely need lots more.
All right.
Yeah. Any boa morphs, we definitely can use lots more. There's an okay amount that's come in over time, but yeah, lots more for sure.
Good stuff. All right. Now garter snakes. They're a little less common, but I know there is interest in the garter snake community for genetic testing for them.
If people have, I don't remember seeing any sent in for garters yet.
Oh, bummer.
There could be, a few that I just didn't realize they were garters, but I've kept everything that people send in, say it's, for research or new testing. So if any of you have sent them in, then message us and say, hey, I sent some in, and I'll find them and make sure we can do something. But if I have any, there's not very many. We would definitely lots more.
Fair enough. Well, if you want to, those of you watching, if you want garter snake shed testing in the future, share with your other garter snake keepers and friends and breeders, and that could become a thing. Pituophis. So that's our pine snakes, like this gorgeous pine. I think that's one of J. Jacoby's females. Really pretty girl. And bull snakes, gopher snakes, all of our loud hissing friends. Where are they at?
We do have a few, but not a ton. So definitely anything people want to have tested for Pituophis, I'll need several more coming in.
All right, fair enough. And let's see, Lampropeltis. So that's a big family. Kings and Milks, all them. I know you have Honduran sheds because you breed some.
Yes.
Yeah.
Yeah, not many sheds that I can remember to be sent in to develop new tests. Nothing pops off in my head where we got like a bunch, but there's probably a handful. But if it's something any of you with Lampropeltis, something that would be a helpful test, get people together, send in sheds in. If all of a sudden I see 20 or 30 for a certain morph, then it will, move up the list real quick.
Yeah, because basically the more sheds from the species that you get, like basically if there are more sheds for it, you're going to prioritize that, right?
Yeah, definitely.
Nice.
Yeah, because that means that we can do it. If you don't have
Exactly.
very
Yeah.
many, yeah, it's not likely to work. But if I've got, 20, 30, 40 of just one mutation, it's very likely we can work with that and make something
Right.
happen.
Fair enough. So what about corn snakes?
Corns people have done a great job, and I think that the genome that's publicly available will be good enough. We'll be able to use that, but we might need to do some of our own whole genome sequencing. We'll see. But that we've had a lot of people send in corn snake sheds. Y'all have done great.
Awesome. Well, that's great. Also, I love this picture. It's from a video that went viral from Justin Dahl of extrasensory photography, who I'm trying to get to do for one of our Why I Do This segments later on. But it was a video of just the biggest male corn snake I've ever seen that he found while herping in Florida. This guy is a hoss. It's amazing.
That is awesome.
Yeah. Let's see. I gotta ask for me just because I like black rats. Have you gotten anything for black rats other than the stuff I sent you?
Not much.
Not much.
That I can remember. But once again, you've sent some in. Feel free to send a message. Say, hey, I did send some in.
Yeah, exactly.
Nothing sticks out in my. So if
All
I do
good.
have some, that's not very many.
Yeah. I may eventually make an actual black rat post, but I really love this picture on the right of that's a leucistic female from Metazotics, who we had on one of our segments a couple of weeks ago. So good picture.
Pretty.
All right. Are there any other morphs that you wanted to give an update on that I didn't have in our slides? Like, or any other morphs or species, I should say?
Species. The only other thing I would mention is we are still interested and plan to work on sex determination tests in some groups as well. So it's not really something we need sheds sent in for necessarily. I've got plenty. So blue tongue skinks are one of the top ones. I've got plenty of blue tongue skink sheds.
That's nice.
That are known males and females. But Heloderma, if you have Heloderma, known males and females, I have a decent amount, but I could use a few more. And then any species of Varanus, so any of the monitor lizards, if you have known males and females, we could use a few more sheds. And in the case like with lizards, where a lot of the time you just have small pieces, you don't have a big full shed a lot of the time. Really all we need is enough shed to cover like the top of a quarter. It's okay if it's in 20 pieces. It just needs to be enough to cover the top of a quarter. So for you green tree python people,
yes.
Before we move on, I've been working with Sean and with Bill Stiegel, and we do have something that we're going to make available for green tree again. It will be that complex test where we need sheds from the sire and either a known male offspring from the sire or the paternal grandsire. So it's going to be kind of a pain and kind of expensive, but at least if you have, those of you that have really important green tree clutches, will give you the option to do some sex determination testing. So I hope this week that I'll have that write-up available. I don't know if it'll be on our website, but I'll at least start sending out to a few people and they can spread it around, but we'll have something. It won't be as good as I keep hoping we can get, and maybe it will be that we'll never get as cheap as I was hoping, but it is the best that we can do right now, and so we'll start making that available again.
Fair enough. I know that we were on the hunt for the single gene test, but there's some research coming out that we can explain more in detail once that's out, I guess. Yeah. All right. So with our next segment, the monthly Morph Spotlight. Now that we have 30 some morphs out, we figured that it might serve us and serve you guys for us to do a little deep dive, talk about the history of each of these morph tests, and give you a little update every month or so, maybe sooner, maybe less. So yeah, let's start with the very first one we came out with, that being the piebald test. Go ahead, take it away, Ben.
Yeah, pretty picture from Patrick and Andre from Chimera.
Yeah.
Of I think it's an ultramel pied, but yeah, piebald was the first mutation. So yeah, I think that they've said this was like 8-10 years ago that they started collecting sheds and working on being able to map the pied mutation.
Yeah.
And as many of you know, some of these morphs, which that was an ultramel pied, ultramel is one of them that has 2 different mutations that make it an ultramel. But with piebald, it's one of the more simple ones where it's just one mutation. It's a base change that the Canadian group published. And so there's a very in-depth explanation of the exact genetic change. Doctors Alan Garcia Elfring and Heather Roffey from McGill University published that a few years back. And in our second episode, we also talked a little bit about how they also worked with another group in Georgia to induce a piebald mutation in an anole lizard. So that's pretty cool. They used that information about that piebald mutation and then moved it into another species. So
it was
very
really cool.
Very cool.
Yeah, I'll link the video up in here so you guys can check it out if you haven't seen it.
Yep, and then these are the people they mentioned in the publication, the breeders that submitted sheds to help them do this work. And being, groundbreaking, very first people that did this, it took some time and they had perseverance and made it happen. That's really cool that they did that. And we had a pied mutation, pied test, pied sequence that we could move forward with, know what gene it's in. Very, very cool. Yes, and then for each of the tests, we'll give it a grade. So we'll say this is an RGI grade of A+. And the only way it's going to get a plus is if there's a peer-reviewed scientific publication to go along with it. So with piebald, we do have that. So that's how it gets the plus. And the reason why it has an A is because we've run over 1,000 known samples. So a lot of those are, when you all send in panels and let us know what genes that they have, then we can use that. If it's a known pied or a known het pied and those match, then we can add that to the number of ones that we know the test has worked properly. In the case of piebalds, that's well over 1,000 by now. We've got lots and lots we've run. And then obviously the Canadian group that designed it, it worked perfect. Well, I think they talk about in the publication one that was questionable whether the person knew for sure if it was a het or not. It didn't map or it didn't test the way that they thought. But other than that one, that was kind of a question. It's been a very, very good test.
Excellent. Well, that's good to know, especially for those who are wanting to use it for breeding purposes. Granted, we've known for a while that it's a solid test, but so does that mean that all of the tests that are out are going to be A+, or is there going to be a different story for each test as we go through this?
Each test will be a little different. By this point, a lot of them will be A's, but very few are A+, because that requires that peer-reviewed scientific publication. The yellow belly complex, and then you have the albinos, which includes albino, candy, ultramel, and lavender albino.
Yep.
And then the hypo. Those are all ones that there are scientific publications for. But most of the others that we have or haven't been published yet. So the best they can get is an A for now. And eventually, hopefully, they'll all have publications. But
yeah. Well, very cool. So that I guess that wraps it up for today, for that segment and for our video. So all of you who tuned in today, thank you for watching. And let us know what let us know what you like. Let us know what you think of this new segment. What morphs you're excited to hear more about if we do these little spotlights and deep dives into how they were produced. You got anything, Ben?
I'm just happy. Like I said, when stuff's working, I'm very happy. Things keep going well. You all keep using the testing, and we keep hearing stories of how it's helping you. And each month that goes by, there's more and more stories where we say, there's animals that got tested that now are proving out and they're breeding, and just love hearing that. Thank you for using the testing, and we'll keep it coming more and more, faster and faster.
You got it. All right. We'll call that a wrap, and we'll see you guys next week. Thank you. And outro in 3, 2, 1.